Foster.
I'm Nate Moore, and we're really excited with our research this year because we were able to identify bacteriophage tortellini as the first member of the P2 subcluster. So we started out with a big pool of phage that we sent in for sequencing, which Kyle's going to talk about a little bit later, and we identified tortellini as P-cluster phage. So this figure up here is a favoritor map comparing tortellini to two other P-cluster phages. And you can see the first half of the genome, there's a lot of purple, a lot of nucleotide similarity in the first half. But in the second half, there's remarkably little. So really the focus of our research was to kind of dig into that second half of the genome and see what was going on. And also, we found between the genes of 30 to 42, 10 genes that were really similar, had a lot of similarity to bacterial genes. So we wanted to look a little more into that also.
Yeah. So as Nate mentioned, we kind of had two focuses for our research. The first was figuring out why tortellini had enough similarity to be considered a P-cluster phage, but why it was different enough to be considered in a new subcluster. And then the other part of our research is kind of the unique methodology that was used to characterize tortellini. So 2 years ago, the phage class from CU Boulder sent in a pool of phage genomes to the sequencing facility. And the sequencing facility pulled out a couple interesting things for us to look closer into. So they let us know that there was a P cluster and an F cluster phage within that pool of DNA, but they didn't know which phage was which. So our classmates Tyler and Paul decided to try and figure out which was the P cluster phage. So the way they went about this is they took a P cluster specific contig provided by the sequencing facility to create a primer that would hopefully help them narrow down from this big pool of DNA which one was this P cluster phage. So they were able to narrow it down to two different phage, tortellini and phage la-di-da. They both had the results that were similar enough to kind of possibly be the P cluster, but they couldn't tell which was which. So in order to differentiate between the two, they used a restriction digest. So if you look at figure 2, this is a virtual digest of the P cluster with what they would hopefully observe. And the left shows the results that they obtained. In particular, you notice that they use EcoR5, which is not one of your standard restriction enzymes for the restriction digest procedure, but they used it because they knew what it would cut for a P cluster phage and they knew what it would cut for an F cluster phage, but more importantly, they knew that those cuts were very different. So when they ran the experiment, they saw that the cuts made by EcoR5 were consistent with the P subcluster for tortellini, and therefore we're finally able to establish tortellini as this P cluster phage. So once we had established that tortellini was in the P cluster rather, we looked more into its physical characteristics to see if they matched with the P cluster. So in figure 3A, we have the electron micrograph of tortellini, which shows the Cyphaloviridé morphotype. And figure 3B, it shows the plaque morphology of tortellini, which had turbid, cloudy plaques, which kind of indicated to us that it was a temperate phage. And actually later, when we did some bioinformatic analysis of tortellini, we found that in this region there are several genes such as an integrase, an antirepressor, a CRO-C1 protein that are also typical of the lysogenic life cycle. Also a note, a little bit of background of clustering. In order to be in the same cluster, phage have to have 50% similarity, and in order to be in the same subcluster, phage should meet a threshold of 75% similarity.
So we decided to kind of look a little more into this back half of the genome. So this first figure here is a bar graph for the FAMs found in genes 30 through 73. And so we wanted to see what clusters were continually coming up related to the FAMs that we found in those genes. And so we got some really interesting results because as you can see here with F, there was 20 FAMs we found similar to the F cluster. And that was almost double what P was at 11. And even N and I were both above P. So that really gave us an idea of some of the similarity that F may have in this back half of the genome, and again how little similarity we're getting with the P cluster. This next figure on the left is a dot plot between tortellini and Perky and A cluster phage, or a P cluster phage, and we kind of got what we expected, where the first half of this genome is a really dense line, which is also shown in the famarator map. There's a lot of nucleotide similarity, and then on the right is a dot plot with tortellini and awesome sauce and F cluster phage that was similar. And so we can see this bit here where there's a good amount of similarity in the second half of the genome that almost fills in a piece that's missing with this piece similarity. So we're able to get a little bit of a better idea about the ancestral history between tortellini and the F cluster. This favoritor map down here just gives us a little better view. It's between tortellini and awesome sauce, the one compared up here, of just the second half of the genome, and it just gives us a little better idea of what exactly the genes are that are similar, and just really kind of points it out well.
So Nate kind of highlighted some of the phage similarity that we saw with tortellini, but another big component of the favoritor map and the DNA analysis was that we saw a large region of tortellini that had high similarity with bacterial genes. So Nate mentioned earlier genes from about 30 to 42. The top blast hits were almost all related to bacteria. So we focused in on a couple in particular, and this figure over here is looking into tortellini's integrase gene. So tortellini's integrase is right here, if you can see it. And when we ran it through blasts, the top result was Mycobacterium canarisins, and had about a 99% identity match and a really great p-value. So we looked a little closer into this relationship, and this figure was generated by a program called ClustalX, which allows you to compare the amino acid sequences of two gene products. So in the first part, part A, we've compared tortellini's integrase gene with the integrase of Mycobacterium canarisins. And to help us read the figure, the asterisks above the letters show when amino acids are similar, and the colors also correspond to this similarity. So if you look at figure A, there's an extraordinarily strong sequence similarity between tortellini and M. canarisins, and this continues through a fair portion of the gene product, and even though it kind of peters off at the end, they're a similar length and have some other similar characteristics. Now, if we compare this to tortellini and Perky, who's kind of been our model P1 phage, you'll notice that there's strikingly little similarity between them at all, and even the lengths of the two gene products are very different. And this kind of helped us figure out some of those characteristics that help differentiate tortellini from that P1 cluster, and justified why we could classify it in a new P2 subcluster. So it's important to note that this integrase is not of phage origin. And we looked into the fam, which is 10145, and there are actually only 8 other members of this fam. And interestingly, none of the other hosts were mycobacteria. So these all came from different actinobacterial hosts, and it was a very unique family that we don't really see very often in Mycobacterium. So this, along with several other top hits with bacteria like Mycobacterium abscessus, Avium, Gordonia, kind of keyed us into the idea that maybe tortellini's natural host or primary host is not actually Mycobacterium smegmatis and might be of a different species. And so kind of a note on our future directions would be to hopefully figure out what tortellini's host range would be, and maybe being able to test some of those bacteria whose genes we found very similar with tortellini. And yeah, so what we can pull from all this is that although tortellini's genome had a high sequence similarity at the very beginning of the genome, in the back half of the genome there's strikingly little similarity, and upon further analysis we realized that there was in fact enough differences in the back half of tortellini's genome to justify the new establishment of the P2 subcluster to place tortellini in. And as Nate touched on, when the FAMs of genes 30 through 73 in tortellini were analyzed, it showed a high family similarity with the F cluster. And that kind of gave us a little insight into a possible ancestral relationship between tortellini as a P2 cluster phage and the F cluster as well. And we went over the bacterial similarities that we found, which can also give us insight into some of that lateral gene transfer history that we assume. And of course, we'd like to acknowledge the Howard Hughes Medical Institute and the C-Phages program for all of their work with this program and programs all over the country. And we'd also like to thank Paul and Tyler for their work on this project too, and the very best of the best, Dr. Guile and Dr. Philman, for being our mentors and the best teachers there are. Do you have any questions?
Yeah.